my name is Lisa Scalfone, and I am a postdoctoral fellow in William Robinson's lab at Stanford University School of Medicine. I'm a relatively new user of SPR and of the ProteOn XPR36 system, which I think demonstrates the flexibility and ease of use of the ProteOn system. So this webinar will show one of the ways that our lab has utilized this technology to compare the binding kinetics of human-derived monoclonal antibodies with a panel of closely related antigens. We are using a one-shot kinetics approach, so the programming and auto-sampler functions of the ProteOn allow us to perform these kinetic analyses for many different antigen-antibody pairs with minimal active time by the user.
Thank you for this opportunity to present the work that our lab has done using the ProteOn XPR36 system to characterize the binding affinities of monoclonal antibodies with their cognate antigens. I am a postdoctoral fellow in the lab of William Robinson at Stanford University, and our lab studies the antibody repertoire in human subjects with autoimmunity and infectious diseases. In the study I'll talk about today, we have investigated the binding kinetics of a panel of monoclonal antibodies that were produced from human plasmablast cells following influenza virus vaccination. And I'll also take a few minutes to discuss the use of heat map software for the display of large sets of kinetic binding data. It's very seasonally appropriate to be talking about influenza right now, in the midst of flu season here. And as many people know, this is a single-stranded RNA virus that causes a common contagious respiratory illness. And the infection can range from causing very mild symptoms to some cases where severe complications and even death can occur. Thankfully, a vaccine is available prior to each year's flu season, and right now is the time to get one if you haven't yet. But due to the variation in virus strains each year, the vaccine is not broadly protective, and it requires a revaccination each year for the strains that are predicted to prevail in that particular flu season. The antibodies elicited by the influenza vaccine are largely specific for the two most abundant surface proteins of the virus, which are hemagglutinin and neuraminidase. And you probably know that these give the H and N in the strain names of influenza. And if you look at this image of the virus structure, you can see the repeating units of hemagglutinin and neuraminidase on the surface where they're available for antibodies to bind and neutralize the infection. When we look at the molecular structure of hemagglutinin, and we're particularly interested in hemagglutinin as it relates to the antibody repertoire, so we know that it's a trimer. There are 3 units that make up the protein structure on the surface of the virus, and we also know that certain portions of the sequence vary from strain to strain, and these differences are ultimately what lead to differences in the binding of antibodies from the hemagglutinin of one strain to the next, and ultimately contribute to the need for annual vaccination. Influenza vaccination, as well as natural infection, induces B cells of the host to produce IgM and IgG antibodies that are specific for the hemagglutinin of the given influenza strains. And this is the basis of immunity and vaccination to influenza, as these antibodies can bind to the virus and neutralize it, preventing a subsequent infection. Our lab is particularly interested in the antibody response that's produced by plasmablasts. Plasmablasts are a population of activated B cells that are transiently increased in number in the blood during an acute immune response, and they are believed to be drawn from both the naive and memory B cell compartments. And we define these cells by flow cytometry as CD19 positive, CD20 negative, CD38 high, and CD27 positive. So When we stain the peripheral blood mononuclear cells, or PBMCs, of a human subject, and in this case we're gating on CD19 positive live cells, so live B cells, if we look at the CD20 negative B cell population, and gating on that look at CD38 and CD27 staining, you can see in this plot of an individual who was undergoing an active immune response that they have plasmablast presence that are CD38 high and CD27 positive. And in this person, 2.8% of their total B cells were plasmablasts. But in a healthy individual under normal circumstances, the plasmablast number is something closer to 0.1%. Of total B cells. And at 1 week following influenza vaccination, we see something like 3 to 5% of total B cells are plasmablasts. So there's a huge increase in number that occurs transiently when the immune system is stimulated. And we like to think of plasmablasts as the window that's open on your computer right at this moment, or a snapshot into what your immune system is seeing. And that's part of the reason that these are very fascinating cells to study. A member of our lab, Yan-Chong Tan, has recently developed a panel of monoclonal antibodies that were derived from the plasmablast cells of a human subject 1 week following influenza virus vaccination. And the characterization of these antibodies that I've done using the ProteOn XPR system is the subject of this webinar. This is the particular machine that we are using, and we've been very lucky to have a supportive environment with Bio-Rad, including training and very helpful discussions about our data analysis that has really allowed us to produce meaningful data within a relatively short period of time. And I think it demonstrates the ease of use and power of the ProteOn system that we've been able to do this. The hemagglutinin molecules that we're analyzing are from the influenza vaccine strain. So each year, the influenza vaccine contains 2 different influenza A strains and 1 influenza B strain. And in 2011, the vaccine contained Influenza A California 2009 isolate, which is an H1-like virus, and Influenza A Perth 2009, which is an H3-like virus, as well as the B strain, which was Brisbane 2008. And we're able to purchase recombinant hemagglutinin molecules that exactly match those of the vaccine in order to study antibody binding. We use amine coupling with a GLC chip, and we bind our monoclonal antibodies as the ligand, going up to 500 RU or RL, followed by using the hemagglutinin as an analyte with serial dilutions, generally starting from 100 nanomolar or up to as high as 800 nanomolar for some of the antibodies with a lower affinity. We can utilize the 6x6 multi-channel setup of the ProteOn to screen multiple antibodies at a time with a one-shot kinetics approach. And in our case, the monoclonal antibodies are the ligand. So we run 5 monoclonal antibodies at a time plus a negative control. And in order to get kinetic data, we need to run multiple concentrations of the analyte. And in our case, the analyte is the hemagglutinin molecule. And so we run these in the opposite direction, 5 concentrations plus a negative control, which allows us to get this kinetic data for multiple antibodies at a time. And because this is amine coupling, we can regenerate the chips with glycine, and we've had consistent results with regenerating and testing the same analyte antibody combinations. So it really allows us to screen as many analytes as possible against our panel of antibodies. Looking at the binding of the vaccine strain H3 Perth, we see a variety of different binding behaviors for our different antibodies, and in this case each graph represents a different antibody, and the lines represent dilutions of different concentrations of our analytes. So, for example, you can see here that flu-15 antibody had no binding, while flu-26 had a relatively rapid binding and slow dissociation. And for something like this, we would go back most likely with a lower concentration of analyte to get better kinetic binding data. To determine the KD, we fit the graphs using the bivalent analyte model, and this is the model that most closely fits with our data. The biologic reason for that being because the trivalent structure of hemagglutinin, although most likely behaves closely to a bivalent analyte. And when we look at a table layout of the binding of our panel of antibodies against the vaccine strain Perth, we see that the majority of the plasmablast-derived antibodies did bind to this Perth strain of hemagglutinin at a relatively high affinity or a low KD. And by comparison, the H1 vaccine strain California was only bound by one of the monoclonal antibodies, flu-21 highlighted here, with a somewhat lower affinity, and the Brisbane strain did not bind to any of the antibodies in our panel. These graphs show binding to Brisbane at concentrations as high as 800 nanomolar, and you can see that there's no response. We also wanted to know how strongly these antibodies bind to the hemagglutinin of closely related influenza strains that were not part of the vaccine. And what we have here is an initial screening. So rather than representing different concentrations, each of these lines represents a high concentration of a different analyte. And you can see that many of the closely related HA bound, although some of them, again, for example, flu-15 didn't bind. But we went on based on the height of the peaks in this initial screening to determine the appropriate concentrations for our one-shot kinetic analysis of the closely related hemagglutinin molecules with our panel of monoclonal antibodies. You can see in the table here the names of the closely related H3 strains that we're screening against, so they're closely related to the Perth vaccine strain. And it should be clear that many of the antibodies we screened did bind to these other strains as well, or in some cases even better than they bound to the vaccine strain. And to just make this more clear to visualize, we like to use a heat map that shows the scale of binding, with black being no binding, and then the colors between blue to yellow going from the highest to lowest KD, or the lowest KD meaning the greatest binding. And you can see in the top row is the Perth strain that we're referencing against, and some of these other H3 strains actually bound more antibodies or even greater affinity than the vaccine strain. So to show some examples of that, we have flu-14 is binding 16 times more strongly to the Christchurch strain than it is to the Perth strain, and flu-22 is binding 36 times more strongly to the Wyoming 2003 strain than it is to the Perth vaccine strain. So we believe that these are the plasmablast antibodies that came drawn from memory. As I said, plasmablasts can be drawn from either memory or naive B cells, and most likely these antibodies were originally produced to a previous exposure to influenza or previous vaccination and were recalled into the plasmablast response upon the current vaccine. And we've also tested H3 strains that are more evolutionarily distant, or their sequences are farther away from the Perth vaccine strain. And in this case, we find that not as many of the antibodies bind to the distant H3 sequences. So these antibodies aren't necessarily broadly cross-neutralizing, but they bind somewhat in relation to the strain that they were induced against. And again, to show the heat map analysis with this greater strain, you can see there's more black on this one because some of these strains don't bind as greatly or even at all to the panel of antibodies. And this is just showing the heat map clustered based on the amount of binding so that you can see all of the binders and non-binders together. And you can see again that in some of the cases, these other strains are binding the antibodies more strongly than the vaccine strain, which indicates that the cells producing that antibody were drawn from memory into the plasmablast response. So I thought it might be useful to give a brief tutorial on how we make these heat maps, and I like to use free publicly available software called Cluster and Tree View that were developed in Michael Eisen's group, and they can be downloaded from the lab website and used by anyone who's interested in making a heat map. To start with, you need your data from the ProteOn, so you can simply copy and paste the values out of your kinetic analysis, and it needs to be saved as a tab delimited text file, and it needs to be in a very specific format. The first thing is to delete all of those DNB annotations. So in my table I had put DNB wherever the antibody did not bind, but that won't work with the clustering software, so it needs to be just a blank space for those. Then just File, Save As, tab delimited text file, and it will come to look something like this, and you can see it needs to be up in that starting in the top A1 position with a label in the first well there rather than a blank, and this will allow the heat map software to work correctly with your file. So save this. Now we'll open the Cluster 3.0 program, and we need to load our data file into this, which we can do just from the menu option here. So you select the data file that you just made, a tab delimited text file, and you can see it appears in the window here. And in the case of KD data, I like to go to the Adjust Data tab and log transform the file so that the colors on the heat map will appear with a greater difference than they would if you don't log transform it. The next step is to go to the K-means tab and check the Organize Genes box and put the number of clusters and number of runs as 1. Then simply click Execute, and it will produce a file that's ready to go into the TreeView software. So TreeView can also be downloaded from the EisenLab website, and this is simply a heat map visualization program. So when we open TreeView, we then need to find the file that we just created in Cluster, open that, and then use the mouse in the left pane to select the tree that it creates. The yellow box will select the tree, and you can see here that we have a heat map that looks similar to what I displayed with my data before, but not quite as nice looking, and that's mainly because of the color. So not only because red and green aren't great for colorblind, but just perhaps you prefer another color, and I like to change the colors. And I like to use blue and yellow, but of course it's open for whatever you like. And then this is the final heat map that you would get from such an analysis, and the image of this can then be exported to whatever display program you like to use. To make a legend, simply make a file in Excel the same way as you did with a real data file and put in the KD values that you would like to have on your legend and transform them in Cluster followed by TreeView in exactly the same manner and export this file. And this will give you a nice legend to go with your file. And because this uses sort of a rudimentary font and everything for display, I like to go ahead and improve the appearance manually so that it looks a little nicer, although you certainly don't need to do that. It's also possible to further cluster, as I've shown here, and in order to do this, you simply go into the cluster program and apply a clustering algorithm that's very simple to do, and then reload that back into the same pipeline that's used to make the heat map in the first place, and that will give you a nicely clustered data, and there are many different options you can use to do that. So I hope that tutorial is useful for those people who are getting very large sets of binding data from their ProteOn analysis and need a way to display it. We found that the Bio-Rad ProteOn XPR36 system is a very powerful tool for comparing multiple antigen-antibody interactions, as we have with our panel of influenza-specific antibodies. The human plasmablast-derived antibodies that we analyzed were largely specific for the H3 hemagglutinin in an individual subject following vaccination with the 2011 vaccine. And the binding data show that many of these cells may have been plasmablasts that were drawn from the previous memory response. And we've also shown how heat map software allows us to clearly display the KD values of multiple antigen-antibody interactions that we get from the 6x6 aspect of the proteon. I'd like to thank everyone for tuning in to the webinar, and also the team at Bio-Rad who has been wonderful to work with, as well as everyone in the Robinson lab, particularly Bill Robinson and Yan-Chong Tan, who developed the panel of antibodies. And thank you for your time.